In this section, I'm going to give an introduction to metabolism and then talk about enzymes. Metabolism is a sum of all of the chemical reactions in the cell. Metabolism consists of both anabolism and catabolism. Anabolism is biosynthesis. Synthesis means to make something or to create something. Bio is talking about something living, so creating something that's part of a living organism. The synthesis of cell molecules and structures is anabolism. This requires energy. And catabolism. Catabolism is breaking the bonds of larger molecules to release energy. If you think about a person and what they do in order to survive, they have to eat. And that's one of the major things that they have to do. So if I were to eat something, I'm going to bring that food into my body. I'm going to digest it. So digestive enzymes inside of my stomach and then small intestine are going to break down that food into smaller pieces. The process of digestion is catabolism. Now, the reason why I'm digesting food is so that I can absorb those nutrients from the diet and use those nutrients in order to build my own proteins and my own lipids and my own carbohydrates so that I can make the structural components of the cells that make me. Building up all of those materials, making proteins, copying DNA, making lipids, making carbohydrates, that's all anabolism. So that's building things inside the body. And that requires energy input. Bacterial cells are very similar. So bacteria are going to release enzymes that digest nutrients out in their environment. They absorb those nutrients and they use those nutrients in order to make their own substances in the cell. So catabolism and anabolism is a part of every living organism, and together they make up metabolism. And metabolism is just the big term that covers all of it. It's all of those chemical reactions in the cell. So here you can see a simplified model of metabolism. So you start with nutrients from outside of the cell, and then from there you're going to go through catabolism. So you have to break them down into their building blocks. Once they're broken down into their building blocks, that releases a lot of energy in that process. So all those bonds that you break, that's energy that can be released. And cells have figured out a way to use certain enzymes to capture the energy in the bonds of adenosine triphosphate or other energy-like molecules. And we'll talk about how energy is produced later on. So catabolism, it breaks down all of those nutrients. Energy is released in that process. Now it's going to use those nutrients in order to make its own stuff, to make its own macromolecules that it's going to use to incorporate into its cell. So it goes through the process of anabolism, and it can make more building blocks. It can make the macromolecules, which eventually goes and makes the whole bacterial cell. Enzymes are catalysts. So catalysts speed up the rate of a chemical reaction without becoming part of the products or being consumed in the reaction. In other words, they're going to increase the rate of the reaction without being affected. So there's still going to be an enzyme before and after and during the reaction. They're just going to speed up the rate of that reaction. These reactions would typically happen anyways, but without an enzyme, they take many thousands of years to occur naturally. So if we have an enzyme there, it speeds it up to the rate of real life so that living organisms can get nutrients from their food products without having to wait an eternity for those food products. Enzymes overcome the activation energy, allowing the reaction to proceed by increasing thermal energy to increase the velocity of molecules, increasing the concentration of reactants to increase the rate of molecular collisions, and adding a catalyst. So, enzymes are able to decrease the activation energy, and that allows the reaction to happen at a faster rate. Some characteristics of enzymes. Most enzymes are composed of protein. Some of them do require cofactors or coenzymes. They act as organic catalysts to speed up the rate of cellular reactions. They lower the activation energy required for a chemical reaction to proceed. They have unique characteristics such as shape, specificity, and function. Specificity, that's talking about a specific interaction with something that it's going to change. And the thing that it's going to change is called a substrate. And the substrate is something that we're going to define again a little bit later. But the word's actually on here a couple times. So substrate, and here's substrate again. So substrates, that's the substance that's affected by that enzyme. So specificity, they're specific to act on only certain substrates. They enable metabolic reactions to proceed at a speed compatible with life. They have an active site for target molecules, substrates. They're much larger in size than their substrates. They associate closely with substrates but do not become integrated into the reaction products. They're not used up or permanently changed by the reaction. They can be recycled and function in extremely low concentrations. They're greatly affected by temperature and pH, and they can be regulated by feedback and genetic mechanisms. Every single one of these points on here is something that you should know about enzymes, and we're going to discuss many of these in more detail as we go through the next couple slides. So how do enzymes work? Well, substrates are the reactant molecules upon which enzymes act. So substrates are modified by the enzyme. Enzymes are going to bind to the substrates and participate directly in changes to the substrate. But again, they don't become part of the products, they're not used up by the reaction, and they can function over and over and over again. I like to see or envision the enzyme like a Pac-Man. So a Pac-Man's like basically a circle with a little notched out mouth. And here's our Pac-Man enzyme. And that little mouth can chomp, 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 and it's not going to be affected by that reaction in any way. And that enzyme will change the substrate in some way. But that enzyme's not going to change. The substrate's going to change. So enzymes are predominantly proteins. Simple enzymes are just the protein part. Conjugated enzymes, also known as holoenzymes, contain protein and some other non-protein molecule. So, an apoenzyme is the protein part. The cofactor is a non-protein part that's organic or inorganic. And a coenzyme is an organic cofactor. And we'll talk more about these cofactors and their importance and what they do in just a little bit. But here you can see a picture of the holoenzyme is the entire thing. Holo, think whole. This apoenzyme is just the protein part, and then they may have cofactors or coenzymes or cofactors and coenzymes together. Enzymes have an active site or a catalytic site. The active site is the site on the enzyme where the substrate fits. So it's the actual site where the substrate binds. It's the three-dimensional crevice or groove that's formed by the way the amino acid chains are folded. Each enzyme has a different primary structure, which produces variations in folding, which lend themselves to a unique active site. So if we draw our Pac-Man again, and we have our chomper mouth, this active site can only fit a substrate that looks like this. If a substrate that looked like this tried to fit into that active site, it would not happen. This is what makes this enzyme specific or gives it its specificity. It's that active site, this location, this mouth location here, that folds around that substrate and binds to that substrate, and that makes it specific. Only certain substrates can fit in there. So this is a picture of enzyme substrate reactions. So here's that enzyme, the protein portion, and then here's the substrate. The substrate is going to fit into that active site. So here's an empty active site, and here is the substrate bound to the active site. When they're bound together, it's just called the enzyme substrate complex. And you can see how this one doesn't fit into that active site, because this is a specific enzyme, which only interacts with this specific substrate. Now the enzyme is going to do something to this substrate, and the products are going to be released. So cofactors, there's metallic cofactors like iron, copper, magnesium, manganese, zinc, cobalt, selenium, etc. They're going to assist with precise functions between the enzyme and substrate. Sometimes they can activate enzymes, they can help bring the active site and substrate closer together, and they can participate directly in chemical reactions. Co-enzymes are much larger than the metallic cofactors. They're organic compounds that work with the apoenzyme to alter the substrate. They remove a chemical group from one substrate and add it to another substrate. They can carry and transfer hydrogen atoms, electrons, carbon dioxide, and amino groups. And vitamins are an important component of coenzymes. So coenzymes are larger, metallic cofactors are smaller, and just made up of these metals and a few others that are not listed here. But together, they're going to help that enzyme function. And the enzyme is not going to function properly if these are missing. So this is a list of some common enzymes, their substrates, and their reactions. You can pause the video and just take a couple minutes to read through this. I won't be asking you any direct questions off of this table, but these enzymes may come back later on in this lecture, and if I do mention them again, I'll be giving you the details that you'll need to know for any test questions. Enzymes can be active inside or outside of the cell. Exo means outside of, or the exterior of. Exoenzymes are going to be functional outside of the cell. So they're transported extracellularly. Extracellular is talking about outside of the cell. They break down large food molecules or harmful chemicals. Endo means inside of. So endoenzymes are functional inside of the cell, and they're going to function intracellularly, predominantly in metabolic reactions. So they're going to help with metabolism of that cell. So exoenzymes are pumped out. They're going to help digest stuff in the environment. Endoenzymes are inside of the cell, and they're going to do like the basic metabolic reactions for that cell. Enzymes can be regulated. So constitutive enzymes are always on. They're always doing their job, and they're always present in relatively constant amounts. So constitutive, they're constantly on. Regulated means that they can be turned on and off as needed. Production is turned on or induced, or turned off or repressed. So you can induce or repress enzymes. Induce and repress will come back when we talk about inducible and repressible operons a little bit later when we talk about genetics. But for now, induced means it's an enzyme that can be turned on. Repressed, it's an enzyme that can be turned off. And this can change in responses to changes in concentration of substrate. If there's no substrate, why would you produce all this enzyme and why would it be active? You want to shut it off if there's no substrate. If there's a lot of substrate, you want to turn it on. So there's enzymes that you can turn on and off as needed. Why do we care about enzymes in general? Well, in general, we care about enzymes because they're going to help our cells do all of the functions that we need them to do. But medically speaking, why do we care about bacterial enzymes? Because that's a large part of what makes us sick. They're considered virulence factors or toxins because they contribute to disease. When bacteria secrete these enzymes, those enzymes have the potential to affect our own cells. And there's some capabilities that they have. Some of them can eat through our collagen. Some of them can eat through fat. Some of them fight off antibiotics. And some of them cause high temperature. So the fact that these bacteria are able to produce enzymes, those enzymes are what make us sick. So we really care about them. We want to know what enzymes they produce, and that gives us a clue of what types of effect that they're going to have on the host or on us when we get sick with those types of infections. Enzymes are extremely sensitive to their environment. When an enzyme unfolds, it's called denaturation. So denaturation is the unfolding of the enzyme. If an enzyme unfolds, its active site also unfolds, and it becomes non-functional. So a denatured enzyme, if we had a normal Pac-Man enzyme, denatured would unfold and unravel and that active site would not look normal anymore, and that active site would no longer be functional. Things that can affect the structure of the enzyme include heat, pH, and certain chemicals. Temperature and pH are extremely important. We have to grow bacteria at a certain temperature because that's the temperature that their enzymes are functional at. If we grow them at a really high temperature, their proteins are going to unfold. If we grow them at a really cold temperature, their enzymes are going to move so slowly that we may not even see a lot of cell division or a lot of growth. There may not be much metabolic activity. pH is also important. Enzymes have adapted to certain pH levels, and as we adjust to this pH of the environment that we grow them in, we can change how their enzymes act. And if there's a variation in their pH, they're not going to function properly. So we have to have their pH at a certain range because that's the pH range that those enzymes are going to be functional at. Disruption causes distortion of the enzyme shape. It prevents the substrate from attaching to the active site, and non-functional enzymes are going to block metabolic reactions, and they can eventually lead to cell death. There's two different ways to inhibit enzymes. So we don't always want enzymes to be functional. We want to be able to shut them off occasionally. And there's two major ways that we do this: competitive and non-competitive inhibition. In competitive inhibition, a molecule that resembles the substrate occupies the active site, preventing the substrate from binding. I'm going to skip ahead and show you the picture of this. So here you see competitive inhibition. Here's the normal substrate, and here's the competitive inhibitor. It competes with the substrate for the active site. If this guy gets in there first, the substrate can't bind, and now this enzyme is blocked. So in competitive inhibition, the inhibitor competes for the active site. If the inhibitor binds, then it blocks that substrate from being able to get in there, and the reaction will not happen. In non-competitive inhibition, some enzymes have two binding sites, the active site and a regulatory site. Sometimes this is called the allosteric site. And the inhibitor molecules are called allosteric inhibitors. It's regulated by the binding of molecules other than the substrate to the regulatory site. Often, the regulatory molecule is the product of the enzymatic reaction, which provides negative feedback that slows the enzyme activity once a certain concentration of product is reached. So to explain this a little bit better, here is what would happen. We have the substrate in the active site, and the regulatory site is not blocked. So we have the substrate go in, and the reaction is going to proceed, and it's going to produce this as a product. So this is our normal pathway of what would happen. Now let's say we have a lot of this product, and there's extra product left over. Well, if we have a lot of product, then we can probably shut down this system. Think about a conveyor belt, and it's going to keep pumping out product after product after product. Eventually, you want to be able to stop that. Otherwise, you're going to have overflow of product happening. So, in order to stop this, sometimes the product can actually bind to this regulatory site. If it binds to the site, binding changes the shape of the active site, which prevents the substrate from being able to fit in there. So, again, under normal circumstances, the substrate would go in the active site, and it would produce this product. If we have enough product, the product is then going to bind in that allosteric site, and that changes the shape of the active site, which prevents substrate from going in. That stops this from continuing when we have enough product.